Hello, everyone, and welcome to today's webinar. This webinar is hosted by Jen and sponsored by Complete Genomics. Today's topic is using multiplexed multi-omics to study spatial heterogeneity in ovarian cancer. Thank you so much for being here. I'm Uduak Thomas, Senior Editor with Jen, and it's my pleasure to be your host for today's webinar. We have a great speaker lined up for you today. We have Dr. Sammy Ferri-Borgogno, and she is an instructor in the Department of Gynecologic Oncology and Reproductive Medicine at MD Anderson Cancer Center. We're going to get started with her presentation in just a few moments, but before we do that, let's just go over some background on today's topic. Now, studying the spatial organization of all cells in the niche or neighborhood of a tumor is crucial to fully understand exactly how cancers progress. Because proximity speaks to cellular activity, the spatial positioning of cells definitely plays a role in modulating clinical outcomes. However, most platforms do not include spatial context in the three-dimensional space of a solid tumor with single-cell resolution. And as a result, they lack information on cell-cell or cell extracellular matrix interactions. So in today's presentation, our speaker will share the results of a study that provides insights into the molecular basis of spatial cell heterogeneity in ovarian cancer. She'll present a pipeline of integrated multiplex, multi-omics, 3D spatially resolved modalities using FFPE gynecological tumor samples. The multi-omics modalities she will discuss include non-targeted mass spectrometry imaging, Stereo-seq, and targeted seqIF. You'll learn how these spatially resolved modalities identify analytes in voxels across serial tissue sections, and how they reveal an integrated 3D spatial map that displays cell identity, activation, and energized status. Now, today's presentation will be followed by a live Q&A session. And so if you do have a question at any time throughout the presentation, please feel free to enter it in the Ask a Question panel on your screen. And with that, I'm going to hand things over to Sammy for her presentation.
Thank you so much, Uduak, and good morning, everybody. I'm really excited to have the opportunity to share with you our latest data. So just a brief introduction for who is not very familiar with ovarian cancer. Epithelial ovarian cancer can be divided into multiple histological subtypes, which are believed to be derived from either the fallopian tube epithelium or from the ovarian surface epithelium. And have, as you can see, different gene mutational patterns. And the most common subtype, and the one that I will be focusing on today, is high-grade serous carcinoma. Most of the high-grade serous ovarian cancer are usually diagnosed as stage 3 and 4, where cancer cells spread to or beyond other body parts within the abdomen, and this will limit the opportunity for clinical intervention dramatically and results in a 10-year survival rate of less than 20%. HGSC is also initially sensitive to platinum and taxane-based chemotherapy, which is usually delivered before or after the bulking surgery. But the vast majority of these tumors usually recur within 12 to 24 months after treatments, and patients will die of progressively chemotherapy-resistant disease. Moreover, new immune checkpoint therapies show little to no effect in HGSC patients. So there is really the need to find new therapeutical targets as well as biomarkers that can predict resistance. So a critical component that mediates ovarian cancer patient survival is the tumor microenvironment, which is primarily composed of fibroblasts, extracellular matrix proteins, endothelial cells, lymphocytic infiltrates, in addition to cancer cells. The tumor microenvironment has been shown to directly affect cancer cell growth, migration, invasion, and chemoresistance, cell-cell interactions, as well as matrix remodeling. And the influence of the tumor microenvironment on patient survival can come from multiple mechanisms, including, for example, hindering of drug absorption, preventing immune clearance of tumor cells, and stimulating paracrine growth factors that can signal cancer cell growth. So in the last few years, we've been utilizing multiple approaches to characterize as deeply as possible the ovarian cancer tumor microenvironment. And I would like to show you a couple of approaches that we utilized, highlighting the lacking of these approaches and what we came up during the last year to fill in the gaps. The first example is our spatial image-omic pipelines that combine imaging mass cytometry, or IMC, location-specific transcriptomics, and deep learning to identify the distribution of the different stromal, tumor, and immune cells, as well as their spatial relationship. So briefly, we utilized FFPE ovarian cancer patient samples, and we designed a panel of 21 antibodies that were conjugated with different metals, and this allow us to incubate the tissue section with a cocktail of these antibodies, so the staining is simultaneous, and then we performed mass cytometry. So after the collection of high-dimensional images, we performed image analysis, and after multiple rounds of phenograph clusterings, we were able to annotate the cell types and evaluate the cell densities in both tumor and the stroma and perform neighborhood analysis. And we utilized this data to perform survival prediction, investigating if the combination of the markers that we utilized in IMC were able to allow to divide the patients into short-term survival or long-term survival. And simultaneously, we also took advantage of microdissected transcriptomes profiles from matching samples, and we correlated the IMC cell densities with the gene expression profiles. So we compared the spatial cell interactions between ovarian cancer long and short-term survival patients and utilized the nearest neighbor analysis for feature selection to predict patient survival by logistic regression. So as you can see, we identify a signature of 11 neighborhoods. So basically, it's two cells that are close to each other, and the combination of these 11 neighborhoods plus the age of the patient is able to, we built a model, and this model is able to discriminate the long from short-term survival with very high sensitivity and specificity. So we also performed correlation between the IMC cell subtypes identified and the transcriptome profiles from the microdissected stromal and epithelium compartment of HGSC. So this was a lot of data, but just to give you an idea, this is a cartoon we generated by combining the microdissected profiles and the IMC phenotypes, and we identified multiple stromal or cancer genes that are correlated with the IMC features and that are associated with either a tumor-promoting or an immune-suppressive microenvironment. So a second approach that we applied more recently, we developed a spatial transcriptomics pipeline that combine region-specific genes from spatial transcriptomics and cell type-specific gene sets from single-cell RNA-seq. And we utilize this pipeline to dissect the heterogeneity of the ovarian tumor microenvironment even further. And we performed also region-specific ligand receptor interaction analysis. So to go a little bit more into detail, in this case, we utilize fresh frozen high-grade series ovarian cancer patient samples, and we performed spatial transcriptomics utilizing 10x genomics sequencing chips. And after a preliminary analysis, we identified the EST clusters and region-specific gene sets. Then we took advantage of publicly available ovarian cancer single-cell RNA-seq datasets, and we performed our own analysis to identify cell type-specific gene sets. Then we utilized multimodal intersection analysis, and we performed a hypergeometric test to assess significant enrichment of cell types in each spatial transcriptomics spot. So, moreover, we performed region-specific ligand receptor interaction analysis only on the tumor stroma interface. So with the multimodal intersection analysis, we were able to segment the tissue into tumor areas and stromal areas. And then we identified those interface areas, and we performed the ligand receptor analysis by identifying either ligand or receptor in the tumor and either the corresponding receptor or ligand in the stroma. So we found a specific subpopulation of cancer-associated fibroblasts that express alpha SMA, vimentin, and PDGFRB. And this subpopulation can confer a more aggressive type of high-grade series with decreased immune infiltration and, as a consequence, reduced survival. From the ligand receptor analysis, we also identified LRP5 and ApoE as a very important crosstalk that happens at the tumor stroma interface, and this crosstalk has a major role in modulating the malignant phenotype of high-grade series and is also associated with decreased survival. So this is a model that we develop after this analysis. So as you can see, in short-term survival patients, there is a huge amount of cancer-associated fibroblasts that overexpress SMA, vimentin, and PDGFRB that surround the tumor cells. Moreover, these fibroblasts secrete ApoE that binds to LRP5 receptors that are overexpressed in the tumor cells. And these mechanical bindery, together with the crosstalk happening between the tumor cells and the CAF, does not allow the immune cells to penetrate into the tumor and basically to do their job of immune surveillance. On the contrary, in long-term survival patients, as you can see, there are a lot less cancer-associated fibroblasts, and there are a lot more mesenchymal cells that express only SMA and vimentin. So due to the fact that there is not a mechanical barrier and the crosstalk between ApoE and LRP5 is a lot less compared to short-term survival, immune cells can more easily infiltrate into the tumor and perform immunosurveillance. So these two studies were really useful for us to understand general information in the tumor microenvironment and the interaction between the different cell types. But clearly there are a lot of missing pieces because we didn't have the possibility to test different analytes on the same tissue sections and basically have a complete picture of what is going on in the tumor microenvironment. So to give you an idea of what I mean, I would like to do a quick comparison. So for example, if you would have to move into a new city, what would you consider in making your decision on where to live and exactly in which neighborhood of the city where to live? Well, a tumor cell, let's say, does the same type of consideration and try to place itself and try to modulate the microenvironment for its own benefits and its own needs. So for example, you would consider the cost of living that can be compared to nutrients and resources, so metabolites, lipids, and oxygen. You would consider the cost of transportation that can be compared to cell migration. If you have to commute to work, you will consider if there is possibility for public transportation that can be compared to vasculature. You would evaluate the climate, if you think it's advantageous or not, so immunosuppressive or not. If you have kids, you would research the school district, which could be compared to the immune cell health. And then you would want to for sure meet your neighborhoods and plan for the future that in a tumor can be compared to growth and metastasis. And eventually, if you have to commute with a car, you will need gas. And so you will have to evaluate the presence of gas stations nearby that can be compared to the microbiome. Well, with the two studies that I just presented you, you can see that there were a lot of information that we couldn't assess. And so we decided to develop a new pipeline that would allow us to consider all of these factors, utilizing the least amount of tumor sections possible, and also contain the cost of the analysis. So we developed a 3D targeted and non-targeted multiplex multi-omics workflow to dissect the tumor immune microenvironment. So we utilize FFPE tissue section, and in particular, we utilize 17 5 microns tissue section that basically correspond to an 85 micron tissue block. So the first and the last section, we performed H&E staining the top and the bottom. And then as you can see, there are 3 layers that are separated by these light blue slides. So these light blue slides were a reserved section for future experiments and validation, while each of these layers is composed by 3 different sections. And these are the details. So on the top layer, we performed mass spectrometry imaging, which is non-targeted spatial metabolomics, glycomic, and peptidomic. On the middle section, this is the only targeted approach that we utilized, and we performed sequential immunofluorescence utilizing a 20-plex antibody panel. And in the third section, we performed Stereo-seq Stereo-seq, which is non-targeted spatial transcriptomics at subcellular resolution. So then we combined this information together, and we basically created a 3-dimensional stacks, and we analyzed these sections, and they were a 3D voxel. So when you analyze images in 2D, you consider a pixel. In this case, we build it up in 3D, so we called it a voxel. So just a few more details on the specifics of and the rationale of this approach. So if you look at panel B, you can see that this is like representing a tissue, and in the colors, there are different kind of cells. So if you think about it, a cell has an average diameter of 15 microns. So if each of our section is 5 microns, means that in 3 sections, we can have the same cell basically, and we can perform different multi-omics analysis on each of the section, and then put the information back together when we performed image alignment. And we can have, in this case, in our approach, 6 different analytes utilizing only 3 different tissue sections. And then the fact that we utilize the 2 sections in between each of the layers for future studies, this will allow us to not repeat the analysis on the same tissue sections, since these 2 layers are going to be 10 microns total. One thing that I would like to point out for who is not familiar with these technologies is the difference in resolution, which is always very important to account when doing multi-omics analysis. For the mass spectrometry imaging, we utilize a resolution of 20 by 20 microns pixel. For the COMET, the resolution is 0.28 microns by 0.28 microns pixel. And for the STOMICS, each pixel is 0.22 microns, and it's separated by 0.5 microns of space. So when we did this analysis at the beginning of the year, we had to utilize bin analysis, which meaning we had to group together a cluster of pixels. So the resolution is a little bit less than the single pixel that you have over here. Right now, in like 3 months, this technology has evolved, and right now we are able to do single cell resolution analysis. So I would like to go into each layer more into details, and I would like to start with the middle layer, which is the only targeted approach that we utilized. So for this, we utilized the Luna-4 COMET technology. So in this case, FFPE tissue section is utilized, and a microfluidic chip is placed on top of the tissue section. And then the staining is performed utilizing 2 antibodies at the same time in different species. In our case, we designed these 20 marker panel that you can see on the right side, in which 3 markers, were the first 3 were specifically chosen to perform tissue segmentations to separate the tumor areas from the stromal areas. And the remaining 17 markers were chosen to phenotype each different cell type that could be present in the tissue. So each cell can be phenotypes, and we can have a specific phenotyping for all of these cell types. So basically, the way the Luna-4 works is that 2 antibodies are utilized at the same time at different species. There is an incubation step, and then an imaging step, and then an elution step, and then the cycle is repeated until all of the 20 antibodies are used. At the end of this experiment, the images are exported in OMNI, and we utilize Visiopharm image analysis software to process the images. So this is a lot of data, but as you can see, for each layer, here I just want to highlight that you can see the 3 different markers that we utilize for tissue segmentation. So keratin in gray, collagen in green, and CD31 for blood vessels in red. And then utilizing the software, we were able to take advantage of these markers to perform tissue segmentation. So if you want to see here in the enlarged image, as you can see, we were able to identify the stroma area in orange, the cyan area, which is the tumor, and then we artificially created the tumor stroma interface area based on our previous data that I show you in our introduction. And so basically, we artificially created this area by eroding the tumor area and eroding the tumor area and the stroma area by 50 microns. So we had a total of 100 micron space that defines our tumor stroma interface. And over here, you can just see the boundaries of the tissue segmentation. So, here is just a zoom in of the larger image, and you can see these lines are the tissue segmentation lines, and you can see the expression of a few of the immune markers that we utilize. So I would like to point out a few examples. So for example, if you focus on the yellow dots, these are CD66B positive cells, which are neutrophils. And as you can see, there is one over here in the tumor area, one over here in the interface, and a couple on the stroma area. But when we put, and this is basically in 2D on only one layer. When we merge the 3 different layers and we perform 3D stacking and alignment, as you can see, there are new neighborhoods that pop up that were not visible in 2D. So as you can see over here in the tumor area, now we have a neighborhood of 2 neutrophil cells. And over here in the interface, there are more neutrophils clustered together and a lot more in the stromal area over here. So basically, this is the first thing that we noticed, that there are like a lot of neighborhoods that were not visible in 2D that we would have totally missed. But when we do the 3D alignment and stacking, we can discover new neighborhoods and investigate further on their biological meaning. So, so we just talked about the targeted approach that we utilized. Let's go to the mass spectrometry imaging, which was our top layer. So in this technology, we collaborated with Dr. Eileen Seeley from UT Austin to develop this pipeline that is quite unique. So from tissue section, basically, it's initially deparaffinized with xylene and then is coated with a particular type of matrix. And then multi-imaging acquisition at 20 microns resolution was performed utilizing a Bruker timsTOF system, mass spectrometer. And then the first data collected were metabolites. Then the same tissue section is further processed, so the matrix is removed, and there is a new matrix coating, which is specifically to identify glycans. And then again, the multi-imaging was performed in 20 micro resolution, and glycans data were collected. And then there was a third round in which, again, the matrix was removed and the tissue section was sprayed with trypsin, and imaging was again performed at 20 microns, and data were collected to identify tryptic peptides from the tissue. And then at the end of all of these protocols, the tissue section was stained with H&E, and these allow us to align all of these images to the one from the Comet Lunaphore technology. So again, this is an overview of all the data we collected from each 3D layer of our study. So we collected a total of 164 glycans, 948 metabolites, and 586 peptides. And so this is just quick zoom in of the data that we obtained. So as you can see, we were able to overlay the tissue segmentation performed utilizing the Comet tissue section. So this was our stromal area, this was our tumor, and in pink is the tumor-stroma interface. So for example, on the left side, as you can see, this glycan in particularly overexpressed in the stromal area, but its expression decreases when it reaches the tumor-stroma interface and is almost nothing in the tumor area. And over here, you can see that there is a little bit of expression of this glycan, again, in the tumor-stroma interface, but not in the tumor. And then on the contrary, this is an example of a tryptic peptide that we identified. In this case, this peptide is very low expressed in the stroma, and its expression increases to reach highest levels in the tumor and in the tumor stroma interface that is infiltrating the tumor areas, but is not touching the stroma one. So these are unidentified. This is an unidentified peptide and an unidentified glycan. So we're now validating further these metabolites and investigating their importance in the ovarian cancer tumor progression by performing MSMS analysis, so we can identify exactly which of these peptides and glycans are. So the third technology that we utilize for this multi-omics approach is Stomics Stereo-seq. So in this case, we performed non-targeted spatial transcriptomics at subcellular resolution. So for who is not very familiar with spatial transcriptomics, I just added a table over here so you can see the comparison between, let's say, the most famous spatial transcriptomics technologies available. So as you can see, for Stomics, there is a spot size of 0.22 microns, and there is a space, an empty space from the center to one spot to the center to the other spot that is 0.5 microns. While as you can just see, for all the other technologies, the dimension of the spots is even is a lot bigger, with the smallest one being 10 folds bigger. So this is mainly the reason why we utilize this technology, because it seems to be the only one that is true subcellular resolution. So briefly, so this technology utilized DNA nanoballs that are beads that are spotted on a chip, and then, as I mentioned, they are spotted every 0.5 microns across the whole sequencing chip. And then the Stereo-seq chips contain also a tail on top of the DNA nanoballs that contains coordinate identity, or CID, for every nanoball. And then at the end, the tail contain a poly-N for RNA capture. So at this point, after on your chip, you are basically gonna able to coat the chip with your tissue section, and then we just follow the manufacturer protocols, in which includes RNA and cDNA in situ, and then we collected all the cDNAs, and we had the unique identities information to retain the X and Y coordinates of each and every spot. And then we utilized the DMB-seq T7 sequencer for complete genomics for the sequencing. So for who is not familiar with this technology, basically we, after cDNA production from our chips, we produced libraries, and we made new nanoballs utilizing the rolling cycle replication technology. And with the new nanoballs, they were spotted on a flow cell, and this flow cell was then loaded into a T7 sequencer from complete genomics. And we sequenced at the sequencing depth of 3 billion reads for each of our tissue section. And the dimension of the chips we utilized was 1 centimeter by 1 centimeter. So in the next step, we collected data and we performed a genome reference match. And in this project, we performed also latent cluster analysis. As I mentioned earlier, we now can do single cell specific analysis for this type of technology, because there is a possibility to stain and image the tissue section and perform image capture that then is loaded into the data collection and into the pipeline for the sequencing, the raw sequencing data analysis. So you can still perform cluster analysis, but it's going to be a lot more details with the single cell resolution. Sorry, I think I duplicated the slide. Give me a second. Yes, so one thing that I would like to point out is the difference between the Stomics Stereo-seq technology compared to all the other spatial transcriptomics technology available, is that the other technologies utilize a poly-T tail that allows you to capture only poly-A RNA within the tissue, while the Stomics Stereo-seq utilize poly-N. What does it mean? That you can not only capture human RNA, but you can also capture miRNA, long non-coding RNA, and also non-human RNA. So you have the potential to see microbiome and viral RNA, as well as mutations and repeated elements with deeper sequencing. So the data analysis for the Stereo-seq was performed in collaboration with Dr. McKee and Shamini from Patomics.io. So over here, you can see the UMAP summarizing the data. So on the left side, you can see layer 1, 2, and 3 of our 3D stack. And you can see that actually, surprisingly, most of the data overlay quite nicely. And on the right side, you can see the latent cluster, the UMAP for the latent cluster analysis. So this is quite a bit of data and very overwhelming. So I'm going to focus only on layer 1, and you can look at the details of layer 2 and 3 from our recent publication. So as you can see over here, this is all genes image represent basically all the genes that were detected with the Stereo-seq sequencing, and then we performed a 3D stack image alignment utilizing the Visiopharm software. So we overlaid the tissue segmentation from the middle section, which was the targeted approach with the Comet, to this image. And so we were able to basically confirm our tissue segmentation and divide the tissue in the tumor, stroma, and interface areas. So, and over here, you can see if you compare these two images that they look quite similar, with the stromal in the middle, the interface, and the tumor areas with also little interface areas. So over here, we just represented canonical tumor and stroma markers for ovarian cancer, like MUC16 and WFDC2, which are overexpressed in the tumor areas. And for the stroma, we chose alpha SMA and collagen that mainly are expressed in the middle stroma area. But honestly, I was not really impressed by the expression of these markers and their correlation with the different tissue sections. So from the latent cluster analysis, we were looking for novel markers that were not previously described and that could give us a better idea of the different subtypes and the heterogeneity in this ovarian tissue. So as you can see, over here, we actually identified 2 new markers, one for the tumor and one for the stroma, that are able to segment tissue in a way better way. So these, in this case, AL357507 is a long non-coding RNA, and as you can see, correlates really well with the tumor areas and is not expressed at all in the stroma and very little in the tumor-stroma interface. And for the stromal markers, we identified IGF-BP7 as a marker that is overexpressed in the stroma area, but not in the interface or the tumor. So I would like to point out a few other examples that were surprisingly to us. So in a few of the clusters of the latent cluster analysis, we were actually able to identify new biomarkers. So in cluster 3, for example, we found 2 new markers that are specific for the stroma area, but they're not expressed elsewhere. And the most intriguing thing about this analysis was what we found in cluster 5 and 13. So as you can see, these markers are only expressed in the tumor-stroma interface, and they were never identified before in ovarian cancer. So a few of these are also potential receptors, and we're not trying to identify if these markers can have a potential role in tumor cells spreading and specifically in the tumor stroma interface, and if they can have a role in communicating between the cells in the stroma, that could be fibroblasts or immune cells or macrophages, and the tumor cells of the tumor area. So just a quick mention of the microbiome data. So as you can see from the Stereo-seq analysis, we had a total of 615 million total reads, and 500 of them were hosts, so were human, and then the remaining ones were not hosts. So among these, there were 52,000 reads that were belong to bacteria, fungi, and viruses. And this is just to give you a quick example that we can identify microbiome-related RNA, and we can divide the microbiome up to the genus level. So data was very exciting, and we're now working to correlate these sequencing data with the spatial data that I showed you earlier in the gene expression profile, as well as the metabolites, which we think is the most intriguing and important correlation, because bacteria can really influence the metabolism and the metabolic profiles of the tumor immune microenvironment. So just quick few more details on the data analysis that we performed. So from the latent cluster analysis, if we focus in this square, I would like to point out that we will have a stroma area, an interface in green, tumor in yellow, and tumor stroma interface also in purple. So we performed voxel analysis. So as I mentioned earlier, when you work in 2D, you analyze pixel, but when you work in 3D, you have to align all of your information from the multi-omics data, and you create a 3D voxel. So in our case, our 3D voxel levels were composed by data from the Stereo-seq Stereo-seq. So for example, you can see here that SMD4A gene was particularly overexpressed in the tumor stroma interface compared to the tumor areas or the stromal area. And then CADPS is another example that was not just overexpressed in the tumor stroma interface in green, but also in purple, which means that it's the interface that infiltrates within the tumor. And then the second layer that we utilized was mass spectrometry imaging. So this is an example of a glycan that was highly overexpressed in the stroma area, decreased a little in the tumor stroma interface, and is very low in the tumor, as you can see in the quantification graph. And then the third layer that we utilized was the multiplex sequential immunofluorescence with the COMET technology, and this was the only targeted approach. So as you can see over here, we just did this example of the raw sequential IF imaging. And then over here, we phenotyped all the cells in the tissue and performed tissue segmentation. And here you can see the example of a canonical tumor and stromal marker that is overexpressed either in the tumor area or in the corresponding stromal area. So I would like to point out that we specifically decided to perform the sequential immunofluorescence, the targeted approach with the sequential immunofluorescence in the middle, and then put the mass spectrometry imaging and the Stereo-seq on the top and the bottom layer. So the reason is because the COMET is the one with the highest level of resolution, and we can precisely phenotype each cell and perform single cell segmentation. And this information can then be translated into the top and the bottom layer in the MSI and the Stereo-seq technologies. So basically, to summarize, we utilize 3 different approaches, and that allow you to use only 3 different tissue sections. In our case, yes, we also perform the 3D stacking, but even if you just have 3 tissue sections, you can basically gain information from 6 different analytes, and this is kind of like the higher level of information that you can gain from only 3 different tissue sections. So in our experience, this approach could provide adds insights into the molecular basis of spatial cell heterogeneity in the tumor ecosystem. A few clinical applications would be we can identify potential biomarkers that can predict overall survival, progression-free survival, and also response to therapy. This approach can also help us to identify new molecular and cellular pathways that can be targeted by repurposed drug combination. And we can also identify underlying compensatory mechanisms that are exacerbated by conventional therapy. In conclusion, this is the most important slide of them all. So I would like to thank all the people that were involved in this project. In particular, my PI, Dr. Mock, Basant and Akshay that performed the COMET analysis, Dr. Burks, that is the director of the Imaging Core facility here at MD Anderson, and is the person that put up with my madness to put together this project. Also, Javier Gomez and Danielle from the Imaging Core were really helpful with the image analysis. Dr. Seeley performed the mass spectrometry imaging part of the story. She was an amazing collaborator from UT Austin. And Trevor McKee and Shamini from Potomics.io helped us with the voxel analysis and stomic data processing. And thank you all for the attention.
Thank you so much, Sammy, for that amazing presentation. Very interesting and informative. And to our audience, we're going to begin the Q&A session in just a few moments. So if you do have a question, please go ahead and enter it in the Ask a Question panel on your screen. We'll be taking questions live for several minutes. Before we begin the Q&A, I did want to let you know that this webinar will be available to view on demand in the next 24 to 48 hours. So please do keep an eye on your email so that you can get the link and you can come back and rewatch the entire webinar plus the Q&A. Sammy, thank you once again. I do have a few questions already here for you. So I'm going to go ahead and jump right in. First question I have markers for hypoxia and acidity been overlaid with various cell locations?
Yes, so we didn't have markers for hypoxia in the targeted sequential immunofluorescence approach, but we identified many of them from the Stomics data sets. And so, so as I mentioned, we performed latent cluster analysis, but that data was overlaid to the sequential IF, so we were able to retain the information for single cell. So it was quite interesting, and it's most of the knowledge on hypoxia. It's very well documented for ovarian cancer, but it was very interested to overlay the hypoxia gene expression profiles with the metabolites and glycans data. So we mentioned some of it in the paper that we just published, but we're actually working to dissect the mechanisms even further in the connection with the metabolic profiles.
Excellent. Thank you so much. Next question I have, why did you choose this particular spatial transcriptomics platform compared to other commercially available ones?
So we've been used in the past a lot, 10x genomics platforms, and they were great. The issue is that they were never single cell resolution. So now there are like a little more higher resolution. They evolved and they have more higher resolution, but at the end, the Stomics was the only one that allowed us to have subcellular resolution, and this was very important for us, especially because with the other technologies, and especially when the tissue is very heterogeneous and you have mixed areas in which you have a few tumor cells, and then it's just like a bunch of immune cells and fibroblasts, and the environment is so crowded, it's really hard to do precise cell segmentation if you don't have subcellular resolution. So that's why we tried Stomics, and we were really pleased with the level of resolution with this technology compared to others, and with the fact that it's non-targeted, because the ones that are very high resolution for spatial transcriptomics are usually targeted, which is fine if you have like specific questions. But in our case, we wanted to be not like have like all the data and not be biased in our characterization.
Excellent, excellent. Thank you. And thank you to our audience members who have sent in questions. Please keep your questions coming in. The next question I have for you, how can such detailed analysis and complete 3D spatial maps influence the scope of future diagnostic tests of ovarian cancer?
I gave a brief example of, like, for example, the CD66B neighborhoods that were not visible in 2D, and we never found out about those in our spatial analysis before. So this could be a good example that you can have new biomarker for both diagnosis and therapy that are not visible in 2D, but in 3D, yes. So of course you would not apply this kind of approach for every single sample, for every single patient that come to the clinic, so because it's going to be really expensive. But the goal is to like narrow down to biomarkers that can be used for diagnosis or prognosis with easier tests, or also utilizing other technologies that allow to 3D scan the tissue block and just easily find, for example, either the morphological structures or just stain the block with specific antibodies for the identified biomarkers and perform diagnosis that way, especially, for example, for like rare subtypes of ovarian cancer that are extremely difficult to diagnose compared to like the more like high-grade series is very common, but there are other subtypes like mesonephric, like adenocarcinomas, and that are very difficult to diagnose compared to the other subtypes. And this can influence a lot the survival of the patients because of the treatment choices that clinicians have to perform. So if we can help them with diagnoses that involve 3D tools, I think that will be very beneficial for the patients.
Absolutely, absolutely. The next question I have, does the metabolites target list include any lipids in addition to the regular metabolites?
No. So for lipid analysis, unfortunately, you'll have to utilize an additional tissue section because the protocol that was developed by Dr. Seeley does not allow her to perform the lipid analysis with the glycan peptides and metabolites that we performed. She actually implemented a protocol, and I think the paper is impressed, in which she can assess all those analytes plus the lipids in a single tissue section, but you need frozen tissue, not FFPE.
Absolutely, absolutely. And the next question I have, how can information about biomarker distribution in 3D be further exploited in the translational context, as for example, in drug development?
Yeah, so for example, we just now started a huge project in which we're comparing patients that either respond or not respond to either traditional chemotherapy or different regimens of immunotherapy. And we did the studies in 2D, and honestly, we found some differences, but we couldn't really pursue them because we got stuck at the first layer of validation, which was in vitro confirmation of our finding of the spatial biomarkers that we found with the tissue analysis. And so, which is very common, and usually also either you got stuck in the in vitro or in the in vivo part of the story. So we're hopeful that with the 3D component, we can identify new things, and it will be easier for us to validate potential new therapeutical targets. And also, with the level of multi-omics, so having these 6 different analytes all at once, we hope that we will be able to fill the gaps of what was there before, because always the problem is like compensation, right? You treat the tumors with a certain type of drugs, and then after a while they just compensate and the tumors reoccur. So we hope that having the profiles of also the metabolites and the microbiome and everything, we can really kind of like closing the circle and like filling all the gaps and see if we can find the real candidates that will not allow for the tumor to recur in a short period of time.
Excellent, excellent. Sammy, thank you so much. This was really great. Thank you for taking time to answer questions, and thank you once again for the wonderful presentation.
Thank you.
Absolutely. And to our audience, that is all the time we have for today. Just to remind you one more time, this webinar will be available to view on demand in the next 24 to 48 hours. So please do keep your eye on your email, and please share the link when it comes through with your colleagues who might not have been able to tune in today, but who would definitely benefit from the amazing information we got during this presentation. Once again, Sammy, thank you. Thank you so much for taking time to answer questions and for the presentation. I'd also like to, I think we have one more question that just came in actually last minute. Do you have time to take it?
Yeah.
All right. Okay, so here's the question. When you say microbiome, does it mean infiltration from the GI tract or in situ microbes in the ovarian tissue?
No, it's actually in situ microbes in the ovarian tissue. So I think like in the last 5 to 6 years, there is a huge amount of literature that came up studying the microbiome and its distribution in tissues that are not colon or like, canonically microbiomes that are microbes that are resident in those type of tracts. So, and there is quite a few papers that are correlating different levels of microbes with response to treatment and how these patients can respond specifically to immunotherapy and everything. So these are microbiomes that can also be present in the GI tract, but some of them not necessarily, and they're not canonical. And there are a lot of papers also showing how diet and like, for example, very rich diet or poor diet can also influence and change the microbiome levels in these patients. So, and since microbiome are huge consumers of metabolites and energy, it's really important to understand if there is a connection between their presence and the different kinds to the speed at which tumor cells can proliferate and also disseminate, and how this microbiome can also influence eventually tumor cells that are present in the environment. So it's a very evolving and new aspect of studying the tumor environment, but I think it's coming up to be like one of the most important ones. So I'm really curious to see what we can correlate at the spatial level with our characterization.
Excellent, excellent. Thank you for taking that question. And I'd also like to thank, in addition to our speaker, I'd like to thank the sponsor for today's event. Thank you, Complete Genomics. And I'd also like to shout out the Gen Multimedia Group and thank them for hosting this event. So thank you, David Mosley, and thank you, Reynolds Gutierrez. And last but not least, thank you to our audience for being here. From everyone at Gen, thank you so much once again, and have a wonderful day.
Thank you. Bye.